Hi class, it's Miss Dunn here, and we left off class talking about gel electrophoresis. So I just want to play you this animation real quick that explains it. So I'm just going to turn the sound off on it so I can talk over it. But basically, remember, DNA is negatively charged, so it goes away from the negative electrode and towards the positive. And you can see here as it moves, the smaller pieces are going to move further than the larger pieces. So the DNA bands are separated by size, and we can see where the restriction enzymes cut our DNA. Okay, so let's just see what it looks like, just so you can visualize. This is one version. That is what the gel actually looks like. So it's sort of this clear piece, and it's tiny little holes here. Just play you a little bit more so you can see. And okay, so when we put the DNA in, the DNA is suspended in liquid, and there's a dye added to it just so we can see what we're doing here. But once it goes through and we turn it on, we'll have to add another dye because it kind of fades. Okay, so you can see the DNA travels through, and that's what it looks like when we actually light it up. So you can see that, we get these positive bands where the DNA traveled. Okay, so it kind of looks like that. And usually on the far left, we run a sample of DNA where we already know how big these pieces are. And so we can compare our samples to the known samples. So this is always sort of the positive control. Okay, don't know what that's all about, but all right. So let's get back to our notes here. Okay, so for our purposes, we're going to be working with plasmids. And let's take a look at this image that you guys have on your sheet. Okay, so this is a particular plasmid here, and I don't know why that cut off there. But anyway, so here's a bacterial plasmid. And if you remember, bacterial plasmids are useful because you can cut them open, insert genes of interest, stick this plasmid back into a bacteria, and that bacteria will then express that gene and make the proteins that you want. So any protein that a scientist needs, bacteria can make it for us. We just have to genetically engineer this plasmid. Okay, now whenever we look at plasmids, very common plasmids that are used by genetic engineers have known sites, okay? And so these right here, like HinD3, RAN2, here's HinD3 again. These are places where this particular restriction enzyme is going to cut. So HinD3 is a particular type of scissors that cuts at this particular spot, okay? So when we did our simulation today, it just was called enzyme 1 or 2 or 3 or 4. Well, now we're giving them names. That's all it is. And instead of looking for the site, we're just going to tell you where it is, okay? So when you look at maps like this, you can see that between this site and this site, there are 450. This BP stands for base pairs. Okay, so that's how many nucleotides long this section is. So total length of the plasmid, you're going to have to add up all these pieces. So 560 plus 320 plus 450 and 600. You get to see my horrendous math skills. 11, 12, 13. Okay, 13, 18, 19. Okay, so it's roughly 2,000 base pairs, 1930 to be exact here. Okay, so now the next question is how many pieces of DNA would result from the combination of this plasmid with EcoRI? Meaning if we actually expose this plasmid to this restriction enzyme, how many pieces of DNA would we get? So imagine you have this circle, like imagine a bracelet or something like that, and you're going to cut it here. Oh, oops, not HinD3, excuse me. EcoRI, so we're going to cut it right here. And there's no other echo sites. So if we cut this circular piece just once, we're only going to have one piece of DNA. All right. Now in number 3, it says how many pieces of DNA would result from this combination of plasmid. with Hindi 3 and Ran 2. Okay, so now we have to imagine we're cutting here, here, and again here. Okay, so we're going to get this small piece right here. Okay, so we're cutting there, so we're going to get this 320 piece. We're also going to get this 450 base pair piece, and then this whole thing. So it doesn't say that we cut with Echo RI, so we're going to just ignore that site because we're not adding that enzyme. All right, so if we do that, 1, 2, 3 pieces will result. So we can put, oops, it's not letting me write there, but 3 pieces for that answer. Oh, it doesn't look like over there either. Okay, so let's move on to this section here. So now what they want us to do is pretend that we're running this through a gel. So these are all the different lanes. And so it looks like our A, B, C, D, E is off a little bit. But let's take a look at A. So let's just say this is A, B, C, D, E. Okay. And so for A, we're cutting the plasmid with Hindi 3. So remember, Hindi 3 cuts in 2 places. Okay, so I want you guys to pause the video, make sure you find where that cuts. And so the first thing you want to do is figure out how many pieces would result. And so I'm going to go back to that picture. So Hindi 3 is right here. So we're going to get this 320 section plus 450. So 320 plus 450 is 770. So this whole section is 770. All right, and then we're going to get this entire piece here. So 600 plus 560 would give us 1160 base pairs from here all the way around. Okay, so if we just cut with Hindi 3, we're going to get those two. So 770 and 1160. Let's just write that down. Okay, now remember how I said in the left lane we always run pieces where we know the length. So this piece right here we know is 5,000 base pairs. This bottom piece is only 200 base pairs. Remember, the smaller it is, the further it can go. And so we've got this 770 piece, so we literally just estimate where that's going to go here. And so it's got to be bigger than 500 but smaller than 1,000, so I'm guessing it's going to be somewhere pretty close to the middle there. All right, so we're going to get a band there. 1160, so 1160, that's more than 1,000, but not 1,500, so it's going to look somewhere like that. So that's what would show up in the gel if we hoped that it would all cut with Hindi 3. All right, let's try figuring out for Echo RI and Ran 2. And you might want to pause the video and try to do it yourself and then check if you got the right answer later. Okay. So Echo RI Ran 2, let's go back here, and I'm actually going to erase some of what I've already drawn. Okay. Okay, it's going to make all this easier to see. All right, so Echo R1 is right here. Going to cut that right there, and Ran 2 right there. Okay, so we're not cutting with Hindi 3, so we can just loop this whole piece together, which means we're going to add 560 plus 320 all the way, oops, all the way across there. So that means it's going to be 880 base pairs. All right, and there's no other Echo or Ran sites, so we're going to add 600 to 450 here, so that's 1,050 base pairs from here to here. Okay, so we're going to get those two pieces, 880 and 1050. All right, so that's going to go into lane B. 880 is, well, that was 770, so I imagine 880 would be a little bit higher, and 1050 is even higher, kind of like right there. Okay. Now the whole plasmid, that means it's not cut with anything. Remember, going back to our answer, that was 1930 right here. So 1930, that's pretty close to 2,000. So that's probably going to be right on the money there. All right. And letter D, a plasmid that has a 500 base pair gene inserted into RAN2. Okay, so let's go back. So RAN2 is right here. So the way I like to note this, and actually why don't I go through and erase this once again. Okay. Okay, so back to this. Now, 500 base pairs are going to be inserted into this spot, which means we're going to cut the plasmid open and put in a new piece of DNA. So the way we like to note this, so sometimes you'll see this, is we just add in kind of a notation like that. So we're adding in this slice of DNA, and so that's going to be 500 base pairs long. So if that insert works correctly, this plasmid should get 500 base pairs bigger. So instead of being roughly 2,000, it's going to be 2,500. And so if we were trying to do that, we would look for a piece about 2,500, so it'd be kind of higher here, okay? Now, let's look at E. 300 base pair gene inserted by EcoR1, then cut with Hindi3. Okay, so now things get a little more complicated. Let's take a look. Oops. All right, so we're going to forget about this, and we're going to start over. And this time we're going into Hindi3. So that's, sorry about that. Oh, I'm sorry, it's going into EcoR1. I was wondering. All right, EcoR1, there's only one slot. That was confusing me because I thought it was Hindi3, and there's 2 spots for that. Anyhow, so 300 is going to go in here, and then we're going to cut with Hindi3. So we're going to cut right there, and what are we going to get? So we're going to get the 600 piece plus the 300 plus the 560. So let's add up all of that. Okay, so this piece is going to be 1460. Plus we have this piece, remember that's 320 plus 450, this is 770 because it contains both of those pieces there. But we're only cutting with Hindi3. And we're going to assume that this 300 piece got in there okay and everything's all sealed up. All right, so we should only get 2 pieces of DNA. So 1460 and 770. Okay, so the 1460 piece is going to be pretty much, that's pretty close to 1500, right? Maybe a little tiny bit smaller. So we'll put that there, and 770 is going to be exactly this piece right here. Remember, we already did that one. So let's just go right next to that and stick that right there. Okay, so your homework is to work on the rest of the problems on this sheet. And basically, everything's pretty much the same type of problem, but when you get to number 8, instead of showing you the plasmid, they are going to show you this. And you're going to figure out what the plasmid does. Okay, so number 8's kind of tough, but I want you guys to try to get through it. And we're actually delaying our lab so you can hang on to your lab sheet. And we'll go over that next class. Okay, good luck on this, and I'll see you next time.